Are you ready?
Yes, I'm ready. It always takes me a while to unmute myself.
That's okay.
Frantically.
Perfect. So welcome everybody to tonight's third virtual Hatfield's Science on Tap. I'm hoping you're someplace comfortable and you have some good food and some good drink as we get ready for tonight's presentation. My name is Cinnamon Moffett. I'm the Research Program Manager at OSU's Hatfield Marine Science Center in Newport, Oregon, and I'll be your host for today. There has been some trouble with the link for tonight's presentation. So if you know of anybody who was unable to join us, in the chat, I just put where the recording is going to go for tonight's presentation. So feel free to share that with anybody who feels like they missed this amazing presentation this evening. A couple of logistics for us before we get started. We are in a webinar format, so that means you do not have a camera or a mic or a screen share capabilities, but we would love it if you would communicate with us and ask any questions along the way using the chat box on the bottom. So if you push that little call out button that says chat, you'll be able to type in your questions. Please do so at any time and we'll answer them at the end. I also just wanted to take a second to promote next month's Science on Tap. So our next event will be on August 19th at 6 p.m. Watch out for a link. We'll try to get the right one out this time. And that event is going to be Julia Parrish. She is a professor at the School of Aquatics and Fisheries Science, also the dean for the College of Environment at the University of Washington. And she's going to be talking about community science in the time of COVID. So that is a very timely event and we're excited to have her join us. If you would like to know more about any of our upcoming events, you can log on to HMSC homepage, scroll to the bottom, and you can find out all the information and the correct links to any of our events there. But for tonight, I would like to introduce Steingass, who is the Marine Mammal Program Leader at Oregon State's Department of Fish and Wildlife, also courtesy faculty at Oregon State's Department of Fish and Wildlife. I always have trouble when I say those two back to back because they don't come out of my mouth the correct way. She earned her bachelor's degree in pre-veterinary medicine and animal science at the University of Wyoming, and she holds a master's and doctorate degrees in wildlife science from Oregon State University. The research she's going to present to you today is a collaborative community-focused project funded by National Geographic Society Research and Exploration Grant. So please welcome Shay. We're quietly welcoming you. Shay, the floor is yours. Take it away.
Okay, well, thank you, Cinnamon, for having me. And thank you to those of you who found the correct link and are in attendance. I hope this is fun for you and something different to do on a Wednesday. So you may have noticed you're not at Rogue Brewery, unfortunately. And so I wanted to have a play on Science on Tap and call this talk Science on Ice because it is science on ice. And I'm going to be talking to you a little bit today about a project that was funded in 2018 by the National Geographic Society for Research and Exploration Grant Program with a number of colleagues. So this is by no means my work alone, but hopefully I can give a nice overview of the story of working with Alaskan communities. So just to make things a little more fun, we will try this out, and if we fail, that's okay too, because it's Science on Tap. So if you want to participate interactively with me here, we are going to do a couple of polls and little Quizlets. So get on your smartphone or on your computer. If you want to pull up another window, you can visit the links or enter the links below there. If you text the phrase, it will text you back a link, and I would recommend clicking on that because that will let you interact within the actual formatted window, which is a little bit nicer. So I'm going to leave this up for 1 minute just so folks can see it. The term lavish ocean was one of the generated phrases they gave me, and I thought it fits, so that's why we went with that. Okay, so I will leave this slide and we'll have more of these that will include the link for you. So a really quick introduction of me, since you can't see me in person. So as Cinnamon said, I'm a 2018 OSU PhD graduate. I'm the Marine Mammal Program Leader for the Oregon Department of Fish and Wildlife, and recently appointed courtesy faculty at the Oregon State University Department of Fisheries and Wildlife. And some of you might be wondering, well, Shay, what are you doing during COVID? And I just thought I would share that I think I'm doing pretty well, really exploring what it means to be a true Oregonian. So this is Ruth, and she is one of our several chickens. And we had a little forest photo shoot. So I've been stuck at home for several months now, and this is what you get. So now I like to think that I'm very lucky to have a calling, a professional calling, and that has become apparent to me through many occasions in my life that are seemingly unconnected, but pretty powerful. So one of my first experiences with the ocean was when I was pretty young, visiting Mazatlan, Mexico with my folks when I was about 7. I was a Wyoming kid, and I remember a lot about that trip, but in particular, I remember looking out of the airplane window and crying as I watched the ocean kind of drift out of my view. I had the same feeling in 2018 or 2019, my gosh, when I got to visit San Miguel Islands with some collaborative work through my current position here with the Marine Mammal Lab of the National Marine Fisheries Service, and got to spend some time in the Channel Islands with a large number of pinnipeds and other marine mammals. And so another interesting experience that I had that was very unexpected was in 2003. That is me on the bottom right there, much younger, when I got to do an exchange internationally to Finland, and we spent a lot of time in coastal communities. And this day was a day in particular where we went to a beach herring cook-off, which is an annual summer festival, and we got to sit on the beach and eat herring stew, which was completely new to me, but a pretty cool experience. In 2005, I got to visit southern India a year after the Southern Indian Ocean earthquakes and tsunamis. And this was a really amazing trip because they took us around to a number of coastal communities that had been impacted by the tsunamis, and they were primarily working class fishing villages. And we got to hear firsthand accounts of how that affected those community survivors. And lastly, a couple of years ago, I got to go down to southern Louisiana and meet with tribes down there who are essentially fighting for their right to protect their land. They aren't recognized, and they're watching their native land essentially sink under the ocean due to sea rise. So those are just some experiences, and I think all in all, what it comes down to with these and with my current position are that working with coastal communities and coastal stakeholders in the marine environment is something that's really special to me. Now, I wanted to start off with our first poll, and I want to see if anyone can get this to work or respond. So again, visit these 2 links, and I'll leave this up for 1 second, and we will then go and see if we get any responses, which I hope we do. Again, so that's Lavish Ocean 303 to 37607. So let's see what happens here. Let's see if anyone is able to do this. I'm going to activate. And what's the first word or phrase you think of when you hear conservation? Got some responses. That's excellent. Leave it up for another 30 seconds or so. As you might notice, the bigger the word is, the more input it has.
Shay, just so you know, we can't see the word cloud being generated.
Oh, you can't? Oh, no. You know what? Let me. There it is. So I think I have to just click next time. How was that?
Yes, we can see that.
Yeah, awesome. Real time. We have environment and we have an environment with a period. Okay, cool. Well, I think that is good. I'm going to go ahead and lock this. And it looks like protection, preserving, environment, future animals, lots of protection. So hold on to those thoughts. Now what we're going to do is go to, I just have to click new share every time apparently. Okay. Thank you guys for participating. It makes me feel like I'm in less of a vacuum here. All right, so a little bit about my current position that I've been lucky enough to have for almost 2 years now. I work with the Marine Mammal Program with the Oregon Department of Fish and Wildlife. We perform a number of different research and management activities throughout the year, and I wanted to highlight a little of the work that we do. You can always visit our website or reach out to one of us to learn more. So we perform marine mammal management in the Columbia River Basin in collaboration with many entities, and that includes the National Marine Fisheries Service, Idaho Department of Fish and Game, Washington Department of Fish and Wildlife, as well as numerous tribes. We perform pinniped ecology and diet studies in the Columbia River Basin, as well as the Oregon coast. We collaborate with the Marine Mammal Lab to study California sea lions and other pinnipeds within the California current system. We have a new unmanned aerial system program that is driven by a couple different folks, and including Mike Brown and Robin Brown, who's my predecessor, and Shay Valentine, to monitor pinniped populations in Oregon. We also work collaboratively with the Marine Mammal Stranding Network to help them out when we can. And we have a number of studies ongoing in terms of marine mammal health and toxicology. But what I'm going to talk to you today a little bit about is something pretty special, and that's something that happened towards the end of my graduate work, but I don't often get to talk about it because it's extraneous to, it was extraneous both to my dissertation as well as my current day job. So this project is working with Alaskan Native communities in a variety of interdisciplinary kind of approaches to studying walrus behavior and ecology, as well as population health of animals and the environment and the humans that rely on them. So today we're going to talk about One Health, which is maybe one of my favorite things. So One Health is the idea that the health of humans, animals, and wildlife and the environment are interwoven and should be considered as such. So One Health is not an easy approach. It requires interdisciplinary research and collaboration. It also requires the consideration of stakeholders and communities beyond just the species that are being studied. And it requires understanding, or at least an attempt to understand, complex links between animals and their environment. So a prime example of One Health is the Arctic communities and native communities of Alaska and the Bering Sea. So Arctic and Alaskan communities have been reliant on Pacific walrus since at least 500 BC. A lot of these villages have also been continually inhabited for the last 10,000 years at least. And in these remote Alaskan villages, natural resources mean a lot more than just recreation. I think we've all experienced lately with COVID what it means to feel particularly vulnerable in terms of your food source and your food supply. And for these communities, this is a constant consideration. So particularly in winter months, persistence of villages and the cultural heritage of the last thousands of years is highly dependent on access to reliant traditional resources. Now, a little bit of biology of the Pacific walrus, which is the study species here. So they are very large animals. They are one subspecies of walrus. They weigh about 8,000 to 1,700 kilograms, and males can be about 12 feet long. So think about essentially the size of a vehicle. Females weigh about 400 to 1,250 kilograms and are slightly smaller. And walrus are benthic suction foragers. So you'll see that was the best image I could get on the bottom left there. They are essentially giant vacuum cleaners that dive to depths of about 300 feet along the Bering Sea shelf, and they suck benthic invertebrates and other prey off the bottom at the margins of sea ice. And so in some unique cases, walrus have also been known to catch certain prey that include small fish. And there is a verbal history as well as recordings of what are known as seal eaters. And these are animals that habitually tend to eat actual other pinnipeds like ice seals. A lot of times these are described as having very oily chests, yellow tusks, and there's something odd about them. Their blubber is often a different color, and these animals are generally not hunted or eaten because there's something just a little bit off about them. So this is a fun little activity here. Our research takes place in at St. Lawrence Island in the Bering Sea. And so what I'm going to go ahead and do is go in here, and I want to see who knows or has an idea where St. Lawrence Island is. So you should get a map image, and I want to see if this works because it's kind of a fun one. Just go ahead and click on the map if you're able to point to where you think St. Lawrence Island is. It's working. All right, these are excellent guesses and excellent map clicking here. So let's go back to the presentation. All right, so St. Lawrence Island is right smack in the middle of the intersection between the Bering and the Chukchi Seas. So you'll see it right in the middle of the map. And what is interesting about St. Lawrence Island is it's right in the middle of the migration corridor for Pacific walrus. So traditionally, walrus in the Bering Sea rely on pretty extensive sea ice throughout winter months. And so during this time, expectant mothers and bull walrus spend the winter foraging off sea ice for primarily benthic invertebrates. Like many marine mammals, winter months are particularly important for gathering energy for springtime reproduction. And so as animals travel past St. Lawrence Island during the expansion of sea ice, then enough animals are taken to essentially subsist and live upon for the next several months. So the males tend to stick around, and then in spring and summer, the sea ice retreats, and females and calves tend to go, as well as juveniles, tend to migrate back north. So one of my favorite things about walrus is how they manage to not expend energy. And walrus migration isn't so much migration like you would expect a whale. They actually just ride the retreating sea ice north and south as it goes. However, in the last decade or so, we've seen some pretty extensive reductions in ice in the Bering Sea. So you will see here 2018, you can see St. Lawrence Island right in the middle of that, and there is no ice. And so what does this mean for communities that depend on marine mammals as well as the marine mammals themselves? So you might look at this and think habitat loss. But it's much more complex than that. So when I see this problem, and you will see an image here of thousands upon thousands of walrus hauled out on a land haul out during a sea ice minimum. So what we see here are opportunities for chronic stress, inter-individual competition for food, behavioral shifts, density-dependent disease, parasite exposure, and perhaps most bleakly or most concerningly is the uncertainty associated with all of that, because these are new circumstances. And so for the communities in the Bering Sea, as you can see here, there is a very big level of fear that a lot of these cultural resources that have been relied upon are disappearing, and with that disappearance, communities' ability to persist and continue to live as they have, as families and communities, may be disappearing. And so Alaskan communities that have been around for tens of thousands of years are now beginning to feel as if they are in crisis. And so what we need to know in these cases is more than just population matrices when we look at stock management. So I'm sure you've seen the headlines that pop up almost every year now, every season. And with drastic changes in the habitat, we start to wonder about things like health factors that might affect walrus. How does the shifting environment relate to changes in the diet? And what are some of the fitness complications? What can local communities tell us about the changes that we're seeing? And this is incredibly valuable to talk to people that actually live in these environments day to day, because no amount of modeling or remote sensing is really going to pick up these really unique patterns. And so lastly, the prime concern too is how do we start understanding this uncertainty? And recently, walrus were not listed as a threatened species, which was a challenge between Alaskan communities and managers. And one of the main reasons that they were not listed is because of the sheer level of uncertainty regarding their populations. And so that means that we need to know more about the biology of these animals in a changing environment in order to conserve them. So a lot of people do research in Alaska, and from my experience, this is what I've seen the typical approaches. So you identify a grant opportunity and define some research questions. Maybe you have some in mind already. Once you've done that and you have a lot of your proposal written, you might reach out to local communities for a letter of support. So they will likely grant you that very graciously, and hooray, you get the grant. Fantastic. But life is very busy, and after you wrap up all this research, often folks run out of time and either forget to do outreach or just don't circle back. And then they move on, and that's understandable. However, it has not historically been a great way to build relationships with folks that are reliant on the resources that are being studied. So this research that we are performing and have performed has a different framework, and I'm very proud of what has been done. It took years of talking to people, attending the Alaska Marine Science Symposium, and really trying to just be in the room and listen. So what we did in this regard is we reached out to people who are reliant on Pacific walrus. We developed relationships with them and did what we could to become aware of the politics and policy and all of the nuances around that before we jumped into the conversation. Working together, we identified some grants that might help answer important questions and concerns, and we worked together to develop proposals, engaging the community from the very beginning of our brainstorming. We continued to engage Alaska Native communities during our research project. And we're currently in the stage of kind of finalizing and delivering findings and thinking about what next steps we can take together. Now, this is a lot up front to work on, but what happens with this is you have an extremely strong foundation to continue to do research in this really remote environment where you very much rely on people. So walruses are co-managed by the Alaska Native communities as per the MMPA, the Marine Mammal Protection Act. And each Alaska community that is a subsistence hunting community for walrus has one representative on the Eskimo Walrus Commission. The U.S. Fish and Wildlife Service is also a co-manager. And so on the bottom left there, you'll see all the wonderful folks that make up the Alaska or the Eskimo Walrus Commission. So a little bit about my colleagues in this project. Dr. Brianna Beechler is at the Carleton College of Vet Med at OSU. She works on host physiology and immunology in regards to disease transmission. Dr. Heather Broughton, who is at OSU Cascades and Bend, and her specialties are anatomy, physiology, and infectious disease ecology. And both these gals are PhDs and DVMs. They have a number of students that they work with as well, and that includes Danielle Schweitzer, Brianna Kaywood, Leslie Cohen, and Rachel Mitchell. So Leslie is a vet student at OSU, and I'm actually happy to see that Brianna just got accepted to medical school to work with Southwestern Native American communities as a doctor, or a doctor in training. So we have this pretty amazing team of folks, and after all of these kind of meetings happened, we agreed upon a sampling plan. And so what we wanted to look at were a number of health factors and fitness factors for walrus. So when animals were harvested, you'll see that little booklet below. We wrote up a little guide, and they did some pretty amazing comprehensive sampling for us on the ice. And that included a collection of gastrointestinal tracts to look at parasites and microbiome, tongue and muscles, so that was looking at parasites, whiskers, which were used for estimating diet with stable isotopes and can also be used for toxicology. And they also collected a tooth for aging, as well as noting stomach contents, sex, and body condition for further analysis. And the cool thing about these samples is they were literally never unfrozen. They were collected on the ice, and they were frozen, and they were shipped frozen. And so they're as fresh as they can be. But that being said, we almost didn't get the samples. So we all were awaiting the message from Alaska Airlines that our package had arrived in Portland. And it was crickets. We did not hear anything. Customer service wasn't super helpful. So I empowered the tool that I could think of, and I tweeted them. So I actually had to tweet Alaska Airlines and said, please help us. Essentially, we have these irreplaceable samples, and if they thaw, then we can no longer use them for their intended purpose. And they got back to me really quick, and believe it or not, they located them within, I feel like it was within minutes or within the hour, and we got them. So it was really exciting. So we have all these samples, and we have a number of research questions of interest. And again, this presentation is a little bit about that story of working with communities. And a lot of the analyses are done or almost done. However, we are now waiting essentially for reopening in regards to finishing up the final analyses. So our first question was the idea that if walrus are, for example, exposed to shifting habitat or other stressors, and they may be consuming different types of prey. And being that these are animals being subsisted upon, we wanted to know if walrus eating fish or other upper trophic level prey, beyond just bivalves and clams, if these animals had a higher diversity of potentially harmful transmissible parasites. And so this was presented by Rachel Mitchell at the Alaska Marine Science Symposium in 2020, right before COVID. And she primarily looked at two very common and important parasites. One is trichinella, and trichinella is a nematode trichinella is the reason that you are told to cook your pork really well. It insists in muscular tissue and is transmissible between an animal being eaten and humans. Secondarily, she looked at sarcocystis, which is another parasite that insists within muscle tissue, and that's also fairly common within pork and beef. And also walrus has been a historic source of this. So Rachel looked at the prevalence of sarcocystis in animals to see if it tracked with body condition. And what was really interesting was for animals that had essentially a higher level of sarcocystis infection, they were tended to be more in the average body type. They weren't unhealthy, they weren't very healthy. And so there must be something going on mechanistically with infection here. And so these are only two parasites, but it did kind of answer the question of if animals appear sicker or are sicker, do they have a higher rate of infection? And it turns out they really don't. So trichinella, fortunately, was not found. And that is good because that means that that is one less thing to worry about. So the second component to this is that question of whether walrus eating fish or other prey that were bigger than clams had more parasite load, and it turned out they didn't. So that's really interesting, and that's within our sample. But what you'll see here is nitrogen values of the whiskers, and we looked to see if animals that had a pattern of higher nitrogen 15 isotopes, which is indicative of a high trophic level, presented more parasitic infection. And that was not the case. So that's within 37 animals, but we can say that it's good to know that despite what animals might be eating, there isn't necessarily a higher risk for infection. And so we use stable isotopes, again, to measure diet broadly. But one component that we don't understand that's very important is the fact that we don't know how fast walrus whiskers actually grow. And this has been one of the most fun side projects thus far. And it's ongoing at several facilities and will be going for the next year at least. So thanks to folks and the walrus at Point Defiance Zoo and Aquarium, Indianapolis Zoo, and SeaWorld San Diego, we have a number of very good-looking candidates here that are letting us measure their whiskers on a weekly basis. So that's being done by husbandry staff on a number of animals. So animals that might be included in this study or are already are Aku and Ginger. These were two-stranded walrus pups that are now at Indianapolis Zoo. We have the very handsome Dozer, who I got to meet at Point Defiance. It is now at SeaWorld San Diego. Middick is at Point Defiance as well. Ukuk is at SeaWorld San Diego, and Pakuk is at Point Defiance as well. So thank you to all those facilities. What we are going to have at the end of this year is a better idea of how to calibrate the whisker values that we're getting. So we sectioned them every centimeter and then analyzed them. But right now we are, and really nobody else knows what that centimeter means. And so just like your hair or your nails grow at a certain rate, whiskers tend to grow fairly consistently with a little bit of variation between individuals. So I'm really excited about this study because it's going to help inform things like toxicology, chronic stress, and of course, diet. So a little fun anecdote is when this study started over a year ago, I took a weekend and drove up to Point Defiance to visit the walrus, because who wouldn't? And I learned a lot about walrus. So one fun thing is that when they are done with you, they don't really move away. They just lay down and roll away, which is awesome. And I also learned a little bit about walrus sleeping habits. So I showed up very enthusiastically, ready to go the next morning, but I had my coffee by 7. I was just waiting to be let in by my colleague, Katie Anderson, who is one of the vets. And I said, okay, well, let me know how to get in. I'll talk to you tomorrow. And all right, so I was awake, I was ready, and I just kind of got a lack of response. Didn't know what was going on. I was worried there was an emergency. And then I got this text message. She essentially said the walruses were still sleeping, and apparently you can't really wake a walrus when it doesn't want to be woken. So she said, well, why don't you come at 9? And I said, okay, sure. And I sent the little walrus emoji, and I was ready to go at 9 and checked back in. And nope, the walruses slept well past 11 that day. So I actually ended up going in like 6 hours later after I'd had lunch. So they're pretty fun animals to work with. Okay, so back to the walrus in the wild. So now that we are working on understanding some things like whisker growth and parasitology, one of the primary questions of interest are what types of potentially transmissible parasites can we identify with microscopic analysis and genetic sequencing? So in the past, it hasn't been super well studied. People have looked for Sarcocystis and Trichinella as they're particularly risky, but genetic sequencing of walrus parasites hasn't really been done. And so students like Danielle Schweitzer looked at microscopic slides, as well as used genetic sequencing to identify microparasites in the large colon. And something that's kind of exciting about this is that we don't know yet, but we will probably have some unidentified species, which will probably be something like a nematode, but it would be pretty neat to discover unique species within the walrus microbiome and microparasite community. So looking at this map on the bottom right, what's really interesting was animals from Gamble, which is the village here on the top point of the island, and animals from Suvunga had very different parasite diversity, which indicates that they are going different places to forage, they're interacting differently. And so there's some sort of population separation, even though they're fairly close together. And another great finding that Danielle found was a small percentage of individuals appeared to carry a majority of the parasites. So you'll see that parasite richness varied between the two villages, or animals harvested by the two villages. And oops, there we go. There's a really cool amount of diversity in parasite communities. So what you see here is a larger graph of relative abundance of different types or phyla of parasites. And so that can include nematode worms or platyhelminthes worms, mollusks, dinoflagellates. And you'll see that not only is the richness super different between individuals, but just the actual composition. And so that tells us a lot about how walrus function as individuals. And I'm sure you've heard about microbiome and how it affects people in so many different ways. And the same is true for wildlife. So with that, we understand more about parasite diversity. And what about just eukaryotes, single-celled organisms of the Pacific walrus? And so another question is, can we start to catalog this? This has not been done either. And so Justin Sanders at OSU Carleton College of Vet Med is utilizing what's called 18S Illumina Amplica protocols, which is simply, it's an RNA or genetic material amplification process and sequencing. So this is, for the first time, we are getting a full picture of the walrus microbiome. And we're also getting the first unprecedented molecular analysis of parasites in sampled animals. So for humans, microbiome has been tied to things like chronic stress, mental conditions, metabolism or body weight, immune function, and general health. And we are just beginning to understand that this is also a factor for fitness within wildlife and can strongly tie into immunological robustness and other things. So Justin's work is right in the middle of being done, and then COVID shut everything down. So it's going to be really exciting to get that work back. So looking back at what we talked about, we kind of visited all of these factors. So we looked at chronic stress a little bit in terms of, we're looking at that. I didn't talk too much about that, but we are looking at cortisol and other stress hormones in animals in relation to body condition. We're looking at diet and inter-individual variation in eating habits and competition. So this is important too, because as you can tell here, it's extremely difficult to get a tag on a walrus. As much as we would like to directly tie movement and diet and health to habitat use, we have to be very careful, because putting a tag out on an animal could cause a massive stampede, essentially. In the future, that might be something that can be done. But for now, what we have are direct inference factors to understand how walrus might be using their environment, what they're eating, and what types of risk factors are out there in relation to what's going to be a very drastically changing environment. So we looked at behavioral shifts, the potential for that through isotopes. We're looking at density-dependent disease through the transmission and diversity of parasites. And again, we're looking at exposure to parasites between individuals and the factor that this can affect communities. And again, uncertainty. What we're finding is the more we're finding out, the less we know and the less work has been done. So this is a small step into a big ocean of research that is waiting. So last component here is nutritional availability. So again, these animals are consumed for subsistence by native communities. And something that's really interesting is walrus meat and raw meat is a significant source of fat-soluble vitamins. So that's A, D, E, and K, as you may have heard. And when you think about it, we spend a lot of time trying to seek out sun, right? Or taking supplements, because vitamin D is something that people tend to get low in. And this wasn't well understood for a long time. And finally, when nutrient analysis came about, it was discovered that raw meat, particularly the meat of very fatty polar mammals, is an extremely bioavailable form of vitamin D. And so for that reason, vitamin D deficiency is never a problem if people have access to consumption of traditional foods. So that's pretty cool. Now, what Brianna did was she looked at isotopes versus fat-soluble vitamins to see if animals that ate differently provided a different level of nutrition for those who might eat them. And that actually did come to be significantly different. So animals that had a higher trophic level actually had a higher bioavailability of some fat-soluble vitamins. So that's important to know, that not only is the food source important for the walrus and their nutrition, but access to the correct food sources also impacts the people that might rely upon them. Now, so we're going to wrap up a little bit here. And this was just a tour of, again, what this project has been like to work with, and all of the questions that we're beginning to answer, even though this first step is really wrapping up. We're finding that just there's not much information out there beyond what we've done. And so now what I want to do is have you go back to the quiz here. And again, you're going to go in and type in what you think of. 1 second, let me share the screen again. When you hear the word conservation. So remember that focus on what we heard before, which was environment, species, protection, etc. So let's see what people come up with now. Thanks, whoever put Shay. That's nice. Couple of smart guys out there. Okay, I'm going to go ahead and lock it. Let's lock it there. Okay, so this is really cool, and I'm almost a little emotional reading this. I'll have to share these results. We went from species, environment, protection, to community, connection, outreach, subsistence. So that's pretty amazing. And I think that's the message I wanted to get across, is connection goes so much deeper than that one charismatic species. And in order to actually conserve species, we need to talk to those who have a connection with those animals. And the #1 proponents for conservation of the Pacific walrus are the communities that depend upon them. So that is pretty darn cool. All right, so let's go back here. Thank you for bearing with me as I switch back and forth. Didn't go as badly as I thought. Okay, so now what is next? And something I wanted to mention and kind of forgot was looking back at those processes of reaching out to communities and in 2 different ways to approach projects. So, you know, I've never called anyone out, but what I will say is I know that a lot of people are using that first method. And the reason that I know that are two things. One, when Dr. Brianna Beechler went to St. Lawrence Island, members of the community asked her, how did you know to reach out to us and ask what we wanted and what we knew? That kind of blew her mind, and it blew my mind, because it intuitively seems to be the right thing to do. It seems to be the right way to approach things. But clearly it hasn't been done in the past, and there's a lot of trust building and back and forth in working with communities, not because they're guarded, but because they have not been listened to or acknowledged in the past. And so I hope we can start to change that and become better partners in conservation. The other reason I know that it doesn't happen very often is that when we attended the Eskimo Walrus Commission this past January, I was really honored and stunned that we were the only independent scientist in the room. And so that was me, representing myself as an OSU courtesy faculty rather than with my agency. But the room was essentially the Eskimo Walrus Commission members who were fantastic and the most engaged and informed audience I think I presented to. No pressure. There was the U.S. Fish and Wildlife Service and Alaska Fish and Game, and then there was us. And that really says a lot, and that says a lot about how they were willing to let us, a few hopeful young scientists, into the room, and I think that's pretty cool. So there were plans for one of us to attend the Spring Hunters meeting in St. Lawrence Island in 2021, but obviously COVID has that a little blurry right now. And we have presented twice to the Eskimo Walrus Commission. We had multiple presentations at the Alaska Marine Science Symposium, and results are going to continue to be presented in multiple formats. So a lot of this work is already kind of done. It's just being pulled together and finalized. And we will make sure to work with communities to distribute that info in whatever way works for them, including flying out there. We will continue to work on publications and distribute our findings. And what I hope is that this can be an iterative project over the years, over time, and maybe not even just us, but other researchers who would like to get involved, I guess, the right way. So it's been a really amazing experience, and I appreciate you letting me share that. So you can learn more about this in these issues. I highly recommend checking out the Arctic report card for this year. There's an amazing and new written report by Bering Sea elders that I hope will be, and I guess that was 2019, I'm sorry. That will be a continuing thing. And I also recommend the book Fierce Climate, Sacred Ground by Elizabeth Marino. She is an ethnographer at OSU Cascades, actually. And it talks about an Alaskan community in the face of climate change. So with that, I really appreciate your attention, and we're doing not too bad on time. So I have several thank yous that I want to get out there. First, I want to thank all of my colleagues and collaborators, particularly Heather Broughton and Bree Beechler, who were also co-PIs on this Nat Geo grant, and we've been partners in crime, getting this thing off the ground for a lot of years now, and it's just been kind of a labor of love, and it's been a really fun side project. This is none of our actual day jobs. I wanted to thank Justin Sanders for his involvement as well, and Sarah Rothenberg, who's working with us to do some toxicology. Brianna Kaywood, again, congrats to her for getting into medical school. Rachel Mitchell and Danielle Schweitzer, who are all working with us, and Leslie Cohen, who did a lot of the whisker work and whisker cutting with us. So thanks to Cinnamon and HMSC and all the parties that are kind of listed here and I talked about throughout, including the zoos and aquaria, particularly the Eskimo Walrus Commission, and the hunters and native villages of Savunga and Gamble, who've been amazing partners. And this wouldn't be possible at all without them. Thanks to the National Geographic Society for supporting us. It was a really big morale booster, as well as the North Slope Bureau and the Alaska Food Policy Council. And so with that, I'm going to go ahead and open it to questions. And you're welcome to email me. And what I thought would be best is if you'd like further resources, I can point you to our projects, some of the things we've put out, as well as the posters that we had at the Alaska Marine Science Symposium. So thank you, everybody, so much. I hope that was a little bit fun.
That was great. And for those folks that are online, we have a challenge that was thrown our way to be an engaged audience. So go ahead and enter any of your questions into the chat box now. And I just want to say, Shay, anybody who gets excited about walrus gut parasites, I love it. I just think that's amazing.
We're all very excited about it.
So we're getting a couple of great questions, and the first one I'm going to summarize a little bit, and it has to do with that slide, the bar graph with the percents, where you were looking at. I don't know if you want to go back to that slide.
Yeah, and can I see the questions somehow?
Yeah, if you hit the chat, they're coming to everybody.
Let's see.
If I pull a pop-up.
Yeah, the chat was hiding. Okay.
Yeah.
All right.
So Gail asked a question about that percent bar graph slide. And wanted to know a little bit more about how that information was gathered.
Yeah, okay, let me go back here. Oh boy, I think I passed it. Sorry, Gail, going on a wild ride here. I may have to just go into the, oh, I think we're getting there. Is this it, Gail? Let's see here. Okay, major algal groups, yes. So this involved, let's see here, primarily large intestine content. And yeah, so it might be partially food intake, that's correct. So this was just genetic analysis of eukaryotic communities. So in terms of the dinoflagellates and other organisms, that could definitely be secondary intake. And what I would actually guess, and something that we thought about, is because the walrus eat bivalves that eat algae, some of the parasites just within the bivalves themselves and the algal materials that they eat are going to show up in that signature. And because they're giant vacuum cleaners, which is pretty amazing, their capacity to suck up food, I'm sure they get a lot of bottom sediment in there. I don't know if I can give too many other details beyond those guesses. That would be something for probably Danielle or Brianna.
Thank you. I'm going to pronounce this next one wrong, but there's a question about morbillivirus, and that's a concern for pinniped species in the Arctic. Do you know if that impacts?
Yeah, so that is something, you know, and I need to see if Justin is working on viruses themselves, but the idea was to do a very comprehensive sampling of microparasites. So that would include viruses and bacteria. I would hope that would be something that would show up if it's there. And again, this is really exciting to do the actual genetic analysis, because in the past it's been largely like microscope gross parasitology observations. So I don't know if it impacts walrus right off the bat, but that's something we can look into. And I think we're going to find a lot of things that we didn't know were there.
So kind of along that line, but looking at the skin, do you know if anyone has looked at the walrus skin microbiome?
Where is that? Oh yeah, so that's a really cool question, Alexandria. That is being done with large rorqual whales right now. There's some amazing studies coming out, and we haven't done that in part, I think, because of that just wasn't a sample that we worked with. But we did have, I think, a pretty large text conversation about how cool it would be. So all of these windows are pretty open. And I'll even go a little further and say that when it comes to walrus in Russia, absolutely almost nothing is known about them, because there just really isn't any research ongoing. And it's very difficult to work across the border in that regard. So there's a lot of really unknown materials. And when I started this project, one of my best, most memorable moments was learning how to use the microfiche machine at Hatfield, because that was the only way I could find the report that I wanted on walrus biology from like the 60s. So there's a lot to be updated. And I think that's why we're kind of excited about the whole thing.
Nice. I'm going to jump one question to ask another parasite question. Can you talk a little bit more about how the parasites that you've been looking at compromise the walrus's health and the health of those that consume them?
Yeah, so that is one of the major questions, is first understanding the full range of parasites that are there beyond the ones that we can just identify visually. And one of the ways that we want to look at whether or not it damages walrus health is look at the prevalence of those parasites, look at immunological function. That's something that Brianna Beechler is doing. And also comparing it against things like body condition or chronic stress, which we can also measure through the Whiskers, for example. So we don't, the short answer is we don't know, and that's because nobody's cared to look at it thus far. But there's definitely implications for the health of subsistence users. In particular, as I mentioned, a lot of those fat-soluble vitamins go away when the meat is cooked, which is kind of a troublesome thing either way. Either you get those nutrients through uncooked meat, but you are potentially exposed to parasites, or you cook it, for example, to get rid of trichinella, but you lose the nutritional value. So we can't give nutritional recommendations, but we are doing that vitamin and nutrient analysis and putting that forth to the Alaska Food Council to hopefully communicate and think about some of these questions.
Nice. And then one of the questions that came from Minda that I thought was an interesting one is, you talked a little bit about the different ways that research has been done. And so she just was kind of asking a little bit more detail about, did this question come from those groups that you were working with, the Alaska community?
Yeah.
Or that process happened to actually inform this specific research?
Yeah, so it was a very symbiotic process. We had some rough ideas. We kind of want to look at parasite diversity and how it relates to diet. And Heather took that to the Eskimo Walrus Commission at the very preliminary phase. And I have to give her kudos to that, because that would have been a pretty stressful meeting when, you don't know exactly what you're doing, and folks want to know who you are. So they came up with a lot of those questions and kind of helped us refine them in terms of what was of concern. And so another thing that we heard from them at the last meeting is we hashed out, well, what are some things that you're interested in now? And microplastics really came up. And again, that was amazing, again, to me, how informed and savvy the audience was, because that was something, for some reason, I hadn't really thought about. But microplastics in Arctic bivalves is a huge issue, and walrus are directly consuming those bivalves. So the next steps that we take are, again, going to be part of that iterative process. They also understand when we present these ideas that it comes down to fundability as well. So it's all within a very flexible framework that includes a lot of brainstorming and a lot of respect and a lot of kind of throwing ideas back and forth and seeing what we can accomplish.
So kind of follow up, I had a question. How is and I know that this is you're just kind of wrapping this up, but have you been able to communicate with those that kind of propose these questions with the results that you have right now, and what kind of feedback are you getting? Yeah,
so a lot of what I talked about today, these graphics, so our students went to the Alaska Marine Science Symposium, and they were able to see Vera Metcalf, who is the head of the Eskimo Walrus Commission, and she's fantastic. So they had some good interactions there. But again, so we went to the Spring Hunters meeting. Brianna did that this past year, flew out to Savunga. We used airline miles to get out there. And then also Heather and I and several students went out to Anchorage this past winter and communicated these results. And they were, I was actually concerned because we had a lot, but they were very well received, and there was a lot of, I guess, overall enthusiasm in the room between all of us about some of these findings and what they might mean. And in particular, the fact that we have the freedom to do this. So, working with Fish and Wildlife Service or ADF&G, they are constrained somewhat in what they can do. And the communities are also constrained in what they can do. And so if we can all rely on each other, we can get some more momentum going and answering questions that people want to know versus what they need to know to perform management actions.
Nice. With that, can you go to your very last slide with your email on it? So we're talking about questions.
Yeah,
so if people have, we're kind of bumping up the end of our time frame.
Yeah.
Yeah, we'll do it that way instead.
Yeah, I'd be happy to answer any questions. But again, I might also point you towards resources. Oops, hang on one second here. Point you towards resources, and then also my colleagues that know we all have our different expertise. And so I don't want to speak for something I am not going to be very eloquent in doing. Is that working?
Wonderful. Yeah, that looks great. You're getting lots of thank yous, and nice to see you again, Shay, and welcome back to Hatfield and all the good things. And hopefully we'll be able to actually see you face to face sometime in the relative near future. But I just want to thank you so much. And for everybody that's online, thank you so much for finding your way to us today and joining us and sticking in there and asking lots of great questions. I hope that you join us next month on the 19th for our next Science on Tap. But if you have any other questions, please reach out to Shay, and she will help you find the right place to get the questions answered. So thank you everyone so much. I appreciate it. And Shay, again, thank you so much for being a part of this and doing another amazing Science on Tap. Shay has done a couple of them for us. First one virtually, but it's been really, really great. And I just am really thankful that you came back and shared with us this amazing work.
Yeah, thank you guys. Thank you, everyone.
All right. Thanks everyone online. Thanks, Shay. I'm going to end the meeting now. We'll see you next month.